Hello everyone, my name is Katrin Clark and my project is about the subcellular localization of Phytophthora pluvialis RXLR effectors during plant infection. The reason we are interested in doing this research is due to a destructive disease in pine trees here in New Zealand called red needle cast, where, as the name suggests, the needles of trees eventually turn red and fall off, which is clearly a problem as this reduces the trees' growth and their production. This disease affects Pinus radiata and also Douglas fir. This issue is of great concern to New Zealand's forestry industry, as a significant amount of New Zealand's total export value comes from Pinus radiata plantations. With that said, it's clearly obvious that in order to mitigate the situation, we need to do research and learn more about the causative agent of red needle cast, which is a type of oomycete called Phytophthora pluvialis. Phytophthora pluvialis, like many other pathogens, utilises effector proteins which are secreted inside the host plant cells and are able to change the plant's physiology or weaken the immune response in order to advance the infection process. I've been focused on a group of effectors called RXLRs. These proteins are defined by the presence of a sequence motif, RXLR, located at the N-terminal domain. There is strong evidence that this region is actually required for translocation inside the host cells. However, the actual effector activity resides in the C-terminus. The aim of my project has been to use fluorescence microscopy to visualize and record the localization of these Phytophthora pluvialis RXLR effectors. I'll just use this.
We'll give you extra time. How are you?
Swear at it. So a couple of steps were done before I arrived. These were the extraction of RNA from the mycelium of Phytophthora pluvialis, and then doing reverse transcription on this RNA to get cDNA. And this is the point where I started from. So I started by doing clonal PCR using specifically designed primers for each effector. I then analysed the PCR products and used I analysed the PCR products, and when I got a successful result, I proceeded to cloning. Two different types of cloning we used. I used gateway cloning and also topo cloning. I then did LR reactions, where I used PB7WGY2 as the destination vector. After the topo and BP reactions, I grew the colonies on selective antibiotic media and then used colony PCR and sequencing to confirm that the effector of interest had been cloned correctly and that it had been transformed into E. coli successfully. Once I did that, I then did LR reactions, followed again by colony PCR to confirm that the reaction and the transformation was successful. Once I had positive colonies, I made mini preps and used those to transform strain AGL1 of Agrobacterium tumefaciens using electroporation. I then confirmed that this transformation was successful, again using colony PCR, and this picture is a example of the result that I got from doing this. And this is for an effector called 18320. And just as is shown in this picture, I then used the positive Agrobacterium colonies to make liquid cultures and prepare an infiltration buffer, and infiltrated around 4 to 5 week old Nicotiana benthamiana plants, which I'd been growing. I then left the plants for around 2 days to allow time for expression, which was under the control of a plant promoter. Once 2 days had passed, I took leaf punches from the sites on the leaf that had been infiltrated and observed these under the microscope. And these are my results. So the video was taken using an epifluorescent microscope, and the images on the right-hand side were taken using a confocal microscope. And the image up to the top right-hand side is showing in yellow fluorescent all of the localizations of this particular effector, and those structures appear to be the nucleus in the middle there, the cytoplasm and some of these smaller structures that could possibly be some type of vesicle. But further work could be done in the future to determine the exact identities of those structures. And that kind of wraps up my project. Thank you very much for listening, and thank you to my supervisor, Claudia Meisrimler, my associated supervisors, Sophie, Claudia, and Arjelin, and thank you to Fire Protection for this opportunity.
Yeah, okay, Katrin, we'll get questions, yeah, and you're very professional when the system works. Stay very cool and calm. Most people pull their hairs out and start having a fit, but thankfully also Sandy was able to resolve it quite quickly. So, questions for Katrin, please. Anybody online want to ask a question? Okay, we've got a question from the audience. Yeah, Jennifer.
I'm just fluorescence seems localized these sort of long ribbon-like structures. You know what this is?
Yeah, do you mean in the video?
Well, in either video or the top picture.
So the ribbon type things is the cytoplasmic streaming. So that's because it's localizing in the cytoplasm, and you can see it just moving along in the cytoplasmic stream there. It's not super strong expression, but I am working at the moment on doing like a kind of control for confirming this using a red fluorescent protein and doing a co-infiltration to see if it's localizing in the same area.
Did you remove the RXLR section of your construct?
Yes. So I was, my primers were made for the effector domain only. So I did cleave, well, yeah, while I was designing primers, I didn't use the RXLR regions.
Thank you very much. Excellent answers to your questions.